will try to give my talk quite fast. So I'm going to talk about the 2D crystallization. So actually the first thing, we are trying to get the 2D crystals of membrane protein. So as you know, everybody, actually what is membrane protein? Membrane proteins are actually inserted or associated into the biological membranes. So the inserted one can be either single pass or multiple pass of membrane proteins. So to make my speak a little bit clear, I basically divided into this entire process up to get the 2D crystals into 4 major steps. So steps 1, 2, 3 and 4, I will go each step in more details. So the first step, actually, we need a membrane protein to get the 2D crystals. So basically, again, it is depend on the what type of membrane protein you are going to work. So if you can get your membrane protein from native tissues or membrane, it will be much easier because you don't have to worry about like a cellular expression system or cell-free systems. So if you cannot get your particular interested protein from native tissues or membranes, so you have to go either cellular expression systems or cell-free systems. So in cellular expression system, normally divided into 2 groups, prokaryotic expression system and also eukaryotic expression system. So again, you have to select which system you are going to use. If you are working with the bacteria, so you can directly go to the prokaryotic expression system. If you are working with the mammalian proteins, so then you have to select the eukaryotic expression system because sometimes you can express mammalian or human protein using the prokaryotic system, but the problem is the transcription and translation processes are different. So you have to pick actually the right system to get your protein. So at the same time, if your protein is toxic to the cells, so you cannot use this prokaryotic or eukaryotic expression system, so you have to go with the cell-free system. So, so at the beginning, you have to spend a little bit time to actually understand what kind of expression system you are going to use and how you are going to get enough protein to get the 2D crystals. So this is very, very important. So I found this review article. Actually, they have reviewed this review article specifically for the soluble protein, not for the membrane protein. But at the same time, you can get quite information, actually, what kind of, because they have talked about so many different soluble protein and expression system, also vectors, and how you can purify, what kind of tag you can include, so many things. So perhaps you can get some detailed information. And yes, it is for the soluble protein, but certain things you can apply for the membrane proteins too. So once you have the membrane protein you are interested, so then you have, because you know that your protein is inserted into the membrane and you have to get out this protein from the lipid bilayer. So you need to select the right detergent. So you need to screen the detergent and then you have to solubilize the membranes and get your protein out from the lipid bilayer. So for an example, detergent molecule has 2 properties. So it has a hydrophobic part and also the hydrophilic part. Once these detergent molecules are exposed into the water or any kind of buffer solution, so it forms a single detergent molecule. At the same time, at a certain concentration of this detergent, it starts to form a micelles or aggregates. So this is very important to consider when you are doing the 2D crystallization process, because you have to remove this detergent in later steps. So if you have like loss of micelles, it won't actually, it will block your process. So you have to consider the CMC, or the critical micellar concentration of a detergent. This is actually the minimum concentration at which micelles are formed. So it's better to have less micelle detergent to solubilize your protein. And because of this property, we normally divided into detergent into two groups, low CNC detergent and high CNC detergent. And also detergent basically divided into three groups according to their polarity: ionic detergents, non-ionic detergents, and sweet ionic detergents. So actually this is the table that I got from our review. So it has quite many lots of detergents. So we divided them into non-ionic detergents, and we put the name and also molecular weight CNC, and also CNC in concentration, aggregation number, and then the average micelle weight. So here I actually highlighted DM. I prefer basically the DM because it has a quite high CNC and it's good to for the 2D crystallization process. For example, sometimes you cannot use the high CNC detergent, so you have to go some low CNC detergents such as DDM or Salmal or some other detergent. So then you have to find the right way to remove the detergent during the 2D crystallization process. So there are more non-ionic detergent and also some ionic detergent and also sweet ionic detergent. For an example, OG is a non-ionic detergent and it has a very high CNC. So that this is also one of the good detergent. If your protein you can solubilize with OG, and it's very good for the 2D crystallization process. So it's again actually this is kind of like a bottleneck in the entire process. So once you have you screen after you screen your detergent, and then you solubilize your protein, and then protein, so detergent molecule will go and incorporate it into the lipid bilayer, and then finally you will have like basically four major components, like a detergent micelles, detergent lipid micelles, and also protein detergent lipid micelles, and the protein detergent micelles. So once you have these things, and then you can go for the further steps. So the third step is the purification, because you extract your membrane protein from the membrane. So this membrane has some other proteins too. So you have to now get rid of the other proteins, and this step is the purification step. So you can choose actually what kind of purification method you are going to use: ion exchange chromatography, size exclusion, or affinity chromatography. For an example, if you use the cellular expression system, so you can add some tags into your gene of your gene, and then maybe perhaps you can use affinity chromatography for the purification. So after you purify your protein, so you can check actually this purified protein is suitable for the later steps. So we call it as a stability test. So you can either run in SDS phage gel or normal native gels, or just simply prepare an EM grid, stain with a negative stain, and then look under the microscope how your protein particles look like, whether you have some aggregates or your particles are monodispersed or not. So for an example, if you purify your protein and if you make a grid, and then you will see some proteins, and at the same time if you see like a big clumps or aggregates, so that means somehow your protein will start to aggregate. So maybe this sample is not good, so you may need to include further purification steps, either size exclusion chromatography or something to remove these big particles. So this is quite important too. So here actually this one is quite homogeneous. I can say more than 80% is good, and you can run a gel, and then you may see very nice single band and also do some gel filtration chromatography, and you have a very nice peak. So here might be some, if you have like lots of aggregates or something, it will come for the void volume peak. So you can do some purification to get nice particles. So if you have this kind of nice particles of your protein, so I recommend, okay, so you can go and do the later steps. So the fourth step is the reconstitution of membrane protein into the lipid bilayer. So in this step, you have to pick the lipids, because now your protein is in a detergent environment. So what you are going to do, you add the lipid and then you remove the detergent and your protein will get and inserted into the lipid bilayer. So choice of lipid also very, very important. So if you can find the native lipids, it will be perfect. But the problem is we cannot get the native lipids, like the protein of you already extract from the native membranes or expression crystals. So, and also you have to consider the phase transition temperature of lipid. This is very important. And also, if you are going to use some synthetic lipids, so handling and preparation of lipid also very, very important. And for an example, for the synthetic lipid, if you cannot find the native lipid, you can go and use the synthetic lipid such as phospholipid, PC, PS, PMOSM. And the DNPC is the most successful lipid so far for the 2D crystallization. So this is quite nice one, but if your protein, you cannot get any crystal from DNPC, actually you can try other lipids also, like a DOPC, POPC, POPG, or E. coli lipids. And at the same time, you can have a mixture of lipids, so you can try. So if you cannot get the native lipid, go with the synthetic lipid, try these common lipids, and if it is work, actually if it is work, or if it doesn't work, maybe you can try a mixture of lipids. Okay, so now you choose your particular lipids, and now the lipids are in the detergent, and your protein of interest also in the detergent. And then what you have to do is you have to remove the detergent, and then this protein molecule will go and insert into the lipid. So then in that case, you will get the 2D crystals. So to remove the detergent, you can apply different methods such as dilution, dialysis, biobeads, cyclodextrin, and there are some other actually methods you can use. But I pick only these four. So formation of 2D crystals, actually it's I don't know exactly how to explain, and then sometimes people believe it's a one-step process, two-step process, or three-step or something. But I found this very interesting article from Rigo Lab. Actually, Mohammed also one of the author in this article, and you can ask even more question about him. And actually, they propose three different methods. So you have your protein of interest in a detergent, and you have a lipid. So it can be like a one-step process. So that means like it form a lipid bilayer, and at the same time, protein will go and insert it into the membrane. And then during the detergent removal, and it form a crystals. And the second proposed mechanism, actually they think like maybe first it form a lipid bilayer, and then protein will get inserted into the lipid bilayer, and then eventually form a crystals. And maybe the third one, it's actually almost like a crystal contacts. So it form again lipid protein together, and then get a nice crystal. But I really don't know which one is happening. I try to actually learn a little bit, but I don't know exactly. Is this a one-step process, two-step process, four-step, or even more something? So this is basically what normally people think about. And I made a very short table about how the dilution and the other processes work on to remove the detergent. For an example, in the dilution, method. So you dilute like everything in your protein, lipid, and detergent by equal factor. But this method is good for high CMC detergent, and there are some structures we already have by doing this dilution method. Dialysis is the most common method. So we use the dialysis membrane to remove the detergent. So in this case, so you have to pick the right dialysis membrane. This is again depend on the size of your protein. And also you have to prepare according to the manufacturer information, because sometimes they may have some EDT or something, so you have to either boil or something. So it's you have to nicely prepare the dialysis membrane, because you use this membrane to remove your detergent. The disadvantage of this process actually, it's good mainly for the high CMC detergent, because high CMC detergent don't produce lots of micelles. But if you use for this low CMC detergent, so you need to give long dialysis time. So in that case, your protein should be able for long dialysis. So first you have to check your protein is at least stable for 1 week or 2 weeks or something. And there are lots of structure and also atomic structure also got from using this dialysis method. So in our lab also we actually got citrate sodium zinc water. I think Fabian, you use the dialysis method, right? And also I use chloride proton antiporter using the dialysis method, and we're quite happy with this method. It's quite simple, and because you can use the dialysis button. If you go to a CC9, we can show you, we have a dialysis machine also, so it's quite nice too. And the other one is the bio beads, and it's polystyrene beads. So you need a very small sample volume, and the problem is these bio beads can absorb lipids. So to avoid that one, what you can do is, so you can incubate the bio beads with an excess of sonicated lipids before you use it. So there are some structures also from the bio beads technique. And the another one is the cyclodextrin. It's a rod-shaped molecule composed of 6, 7, or 8 sugar molecule, glucose molecule. And it's quite nice because if any kind of detergent, it doesn't matter whether it's anionic, supraionic, or non-ionic. And also, it also doesn't matter either low or high CMC detergent. So that means you can use a variety of detergent in this method. And it has a high affinity for detergent than for the lipids. So that means it won't change the LPR during the reconstitution process. So there are some structures using this cyclodextrin. In our lab, actually, we have a cyclodextrin robot also, so you can look around that one also. So once you have your 2D crystals, so you have to see whether you have a crystals or aggregates, or what kind of crystals, the shapes of the crystals and the degree of order of the crystals. So you normally basically use the electron microscope. So negative stain technique we use, and also you can use like a transmission electron microscope at 80 or 120 kilovolts, and also you can look the magnification around 2000 to 50,000. And some laboratories has actually automated systems, so you can load maybe several grids at a time, and then program will automatically pick where is your crystals or something like that. So then you will see either single layer crystals or tubes or multi-layer crystals. So I will show you, like, sometimes you may see just lipid vesicles. So that means your crystallization process won't get nicely, because you only can see some lipid vesicles or some protein aggregates also. So at the same time, you can see some tubes or vesicles, or sometimes sheets or something. So, and or maybe tubes again. So huge vesicles or some sheets. So it's depend actually of your protein, or whether they like to form sheets or tubes or big vesicles. And these are the actually I really like this the first three references. Maybe you can get more information also. And I also include our latest reference. It covered more or less entire to the crystallization processes and everything. So that's it. Thanks very much. If you have a question, you can ask, or maybe we can talk later.